Hello, my name is Lorenzo from Herbert Consortium, which is a Marie Curie initial training network that gathers experts from several laboratories across Europe. Our group works here in Finland and in Sweden to study the most common type of mental illness, which is depression, characterized by alteration in thinking, mood and behavior. And now we are going to tell you our research. Please follow us.
We perform behavioral tests in mice previously exposed to stress factors. Long-term exposure to stress is believed to lead to clinical depression in both rodents and humans. For instance, we can observe the swimming behavior of mice in a glass of warm water. We score the time the mouse spends moving. When it stops swimming actively and makes just enough strokes to avoid drowning, it is defined as immobility behavior and is an accepted correlate of depressive behavior in rodents. We mainly look at the hippocampus region in brain. This structure is involved in emotional responses. From hippocampus, a whole circuitry of communication arises toward other brain regions such as prefrontal cortex, amygdala, nucleus accumbens, and ventral tegmental area, all active areas of research for involvement in depression. We also use viruses to genetically engineer newly born neurons in the hippocampus. The virus expresses a green protein that labels all infected neurons. The same virus can encode an inhibitor of a molecular process in the cell. This approach helps us to find out if that process is important for neurogenesis to occur and for depressive behavior in that animal.
I'm Silvia Ortega Martinez and I'm from Toledo, Spain, even I'm working in Finland at Eleanor Coffee Lab. My main interest in this project is to elucidate the role of adult hippocampal neurogenesis on depression. Birth of new neurons in adult brain is known as adult neurogenesis, and this process is disturbed in animal models of depression. Loss of new neurons in the hippocampus dentaillus is thought to be most important. Birth of new neurons in the dorsal hippocampus is believed to contribute to spatial learning and memory, while birth of new neurons in the ventral hippocampus influences contextual learning and emotional responses such as anxiety. Indeed, there exists a neurogenesis hypothesis of depression, which posits that reduced neurogenesis in the hippocampus contribute to depressive behavior. To understand in molecular detail how the process of neurogenesis occurs in brain and what factors control it, our group studies adult hippocampal neurogenesis in mice. In this case, we look for those changes in neurogenic cell population that are related to depressive behavior. The route to neurogenesis generates new neurons from precursor cells. We use fluorescence markers to detect these cells at different stages along this route, for example as neural stem cells, neuroblasts, or mature neurons. In addition to studies in animals, we perform primary cultures of neural stem cells to study neurogenesis. These minimize the number of animal use and replicate neurogenesis in vitro.
The neurogenic changes that we study contribute functionally to neuronal activity in the brain. This in the end can lead to changes in the way neuronal cells communicate with each other. Neurons release chemicals from their axon terminals that are crucial for synaptic transmission. These neurotransmitters bind to protrusions known as spines. Spine morphology gives us clues about how the spine will deal with the incoming signal. In depression, where patients experience chronic anxiety, there are fewer spines in the hippocampus. To understand what molecules cause this to happen, we study neurons grown in an incubator. We can genetically engineer them by transferring DNA encoding proteins such as green fluorescent protein. This is achieved using spatial lipids. We can easily detect GFP using a laser scanning microscope, and high-resolution images from neuronal dendrites can be acquired and dendritic spines can be visualized. Reconstitution of these images is done using customized computer software, which helps us to precisely measure fine details of dendrites and spine architecture. In this way, we can add or remove genes and discover what molecules are critical for shaping synapses and indeed for regulating their dynamics. Little by little, we start to build a picture of how various molecules cooperate to exert changes at the synapse, and most importantly, we found out which molecules are deregulated in depression and developed drugs that will rescue this loss of function.
In Lund, we use mass spectrometry as one of the techniques to study depression. This technique consists in extracting protein, and in our study we use mice tissue. Depending on the proteins expressed in the sample, we are able to define and study mechanisms involved in depression. Our aim is finding, using mass spectrometry technique, new biomarkers involved in brain mechanism which could be targeted using drugs to reduce depression and improve cognition.
Advanced mass spectrometry, such as cross-linking related MS, is used for deep analysis of not stable protein complexes and understanding of the mechanism in cells leading to depression. During this kind of experiments, the proteins in the complex are linked together, and we can make a conclusion about the protein-protein interaction in the complex based on the mass spectrometry data. This information can help us to design the active molecules able to restore the healthy functioning of neurons.
The genome-wide functional perturbation systems approach will help us to identify novel players in neurogenesis, potentially revealing depression-driving genes. Among our tasks is the development of new tools based on advanced optical methods in order to identify protein complex interaction in the functionally perturbed neurons. This allows us to generate new reporter systems and finally correcting aberrant signals in neuropsychiatric disease, targeting critical signaling pathways with new designed drugs. Undoubtedly, our research will elucidate more about neurogenesis, helping Herbert to describe human depression and anxiety states associated with cognitive decline.